So in this video, I will be discussing glycolysis and the exact process of glycolysis. So, glycolysis. So it occurs in the cytoplasm of the cell. And so overall it splits up one glucose to make two pyruvic acids and two net, keyword net, ATP. So now let's talk about the process. Let me clear this. So here's a good diagram of glycolysis. And so essentially the first step here is the investment step. So you have to invest 2 ATP to get the process going. So you invest 2 ATP to convert glucose into fructose diphosphate. And then it splits up into 2 p-gals. And then, as you can see, NAD plus comes in, gets reduced by electrons, and then so the NAD then forms NADH, and then the ADP, there's 2 ADP to form 2 ATP, and in the end you get pyruvic acid. So essentially you invest 2 ATP, and you end up with 4 total ATP. So you net 2 ATP. And so it's important to remember that glycolysis does not require oxygen. So it does not require oxygen to occur. And some of your biology classes might cover this, but the ATP is produced by substrate level phosphorylation. So it's essentially just a different type of phosphorylation. And so now, remember that glycolysis occurs in the cytoplasm. So the pyruvic acid, if there's oxygen, which is what we will be talking about most of the time when there is oxygen, it will move into the mitochondria to the preparatory reaction. But before we do that, let's just get an overall summary of what happened in glycolysis. So let me clear this. And so you have a glucose, C6H12O6, plus 2 NAD pluses, plus 2 ATP, plus 4 ADP, and 4 phosphate groups, so PI. And then you get 2 pyruvate plus 2 NADH plus 4 ATP plus 2 ADP. And so essentially this right here got phosphorylated to turn into those 2 ADP. The phosphate group was removed to make that. These two combined, ADP and phosphate, to make this 4 ATP. So you net 2 ATP once again. And remember, this is for 1 molecule of glucose. And so now let's talk about the preparatory reaction. Let me clear this. And so as I said before, it occurs in the mitochondrial matrix, in the mitochondria. And what happens is the pyruvate is converted to a 2-carbon molecule. Remember that pyruvate before was a 3-carbon molecule, and so essentially a carbon is removed. And I'll get into the whole process in 1 second. And so here we have 2-pyruvate plus 2-coenzyme A to get, I'll just draw that down there, and pyruvate dehydrogenase is an enzyme that catalyzes this reaction. And also here you need 2 NAD pluses. Whoops. Clear this. 2 NAD pluses, and then that essentially gets turned into 2 NADH. And then in the end you get out 2 carbon dioxides as well as 2 acetyl-CoAs. And this enters the Krebs cycle, assuming there's oxygen. And so this pyruvate, a 3-carbon, gets turned into 2-carbon, and then the other 2 carbons are right here and get turned into CO2, carbon dioxide. And finally, one last thing I forgot to mention. I'll erase this right here to write it briefly. Pyruvate, obviously, it has to enter the matrix of the mitochondria, and so the pyruvate slash hydrogen symporter. Remember, symport means 2 things going in the same direction. Brings it in. And by it, I mean pyruvate.